sociology for 2 years. Then he realized that he liked more mathematics and physics. Maybe he didn't realize that mathematics and physics would contribute so much to sociology now. So it's still time to go back to sociology. But then he studied mathematics. When he finished his study, he moved for personal reason to work in a high school for 10 years. And at that time, he had 3 kids. 2 of them are physicists. The other one is a biologist. And 2 of them are with us today in the talk. So I are part of the last publication as well. Then he did a PhD in mathematical physics and computation of thermodynamic formalism from the University of Zürich. He got the habilitation from the University of Bern, although he was not there, in 1996. And in 1997 he got the bioinformatics position and created the joint group between the University of Zürich and ETH on bioinformatics. Then he got the habilitation in theoretical physics from ETH in 2019.
I don't remember.
And then in 2007 he got the...
This was when I started to go into Alzheimer's research.
Well, so in 2007 he got the full professor position in bioinformatics in ETH. So thank you very much, Ruedi, for being with us.
So thanks for this warm welcome, and I feel a little bit like home. Also, I must say that I envy you for this great building and installation that you have here, and also this institute that would not be possible to such an extent in Switzerland. These complex people are distributed all over in Zürich. This type of vision is really lacking in my home country, and I would like the young people to appreciate this, given all the situations that problems that you sometimes have. You are in a great place. First of all, the sun is shining. Second, you have a great institute, and you have great professors, and this is what you can have in the most rewarding case that you can meet. Okay, this is sort of not my focus of research, but this is a sort of a byway that I do, and you will see other byways that I have in my research. This was actually a study that I started with an undergraduate student, Victor Sase, a German, Tom Lorimer, he's from Tasmania. And my youngest son didn't get the PhD position, and I was quite happy because it was really fun and great to work with him for half a year only. Unfortunately, now he's at the University of Basel. Then we meet a biology expert, and this was my daughter, Rita, and then an old friend of mine, Clemens Wagner, who was one of my first students in Bern, that contributed to the methods that I use here. And the question that I would like to answer is why the brain's wiring might use more than one decay scale, in contrast to the critical network theories that we have read about. Now, the fundamental question, of course, is what is so special about the human brain? We think that we are humans because of our brains, although the brain sometimes is absent, seems to be absent with some people. And the question goes back to the 19th century, when in Berlin, not air hubs were created, but people like Müller, Dubois-Reymond, and von Helmholtz first found out that the neurons are electrically excitable, and this predictably affects the electrical state of the connected neurons, and this means that sort of neurology goes into the physics part of research, and that it may be worthwhile to really think about it. And von Helmholtz then started also his theory about hearing, and you cannot really work in that field without citing him. There was another line of research, all connected here to Spain. Colquier, Ramon, and Cajal, they saw that the human neocortex has a particular layout, sort of a plan, and that in this plan the most striking features are columnar structures that have 6 layers. And now the question emerges from these 2 elements: to what extent neuronal physics and the cortical architecture could explain why we are humans and why we are so much better, as we believe, than animals in doing certain things. And of course, this can only be answered in a context limited by Gödel's theory, and this is also clear that people who claim that they can explain everything from inside the system really don't know about this. Now, the early drawings from Ramon y Cajal, they look like this here. So here you have different types of neurons, here you have different layers. So as we will see, layer number 4 is the input, the basic input layer from the sensory system. Then from here there are many connections to neighboring columns, and here from layer 5 and 6 you have connections to the motor system. And of course this is reminiscent of the 2 hidden layers in neural network theory. So you can think of sort of an inner side of the neural network that relates to your body positional system, and the second one that relates to the outside system, and the both systems need to do computation. If I make a hit movement to his face, he first has to calculate where the movement comes from, and then he decides not to react at all, but to hit back. So this is another picture of this biological data. Here's the cortical surface with the different layers. And by abstraction, this is now a more modern view of the same stuff, but I would not say that this is more insightful than the old Ramon y Cajal drawings. And this is sort of the model that you can have. You have here the layer number 1 that does not really contain a lot of things, then layer 2 and 3, layer 4, 5 and 6. And here these pathways, they just explain or draw what I was just talking about. Now, when you look at biological networks, then this is some biological data that's completely different from the wiring in the neocortex. It's actually the graph of courtship of Drosophila, these fruit flies, when they want to have sex. It's like with the humans. They first have to invite the girl for the movies, then next time they invite them for the movies, then they go and have a cup of coffee, and then they do it an infinite time if they are unlucky. Otherwise they will be allowed to go upstairs with the girls at some point. And so this is all about behavior, and this behavior consists of different acts. And now you can come, if you come from dynamical systems and mathematics, what you want to do is to have a symbolic dynamics consisting of symbols that are completely independent and cannot be decomposed and have no obvious functional ties between the elements, so that you can freely, in principle, combine them. And this is the data we get, and if we now make this courtship graph as this graph of these networks that people usually do, then what you see is two scaling behaviors. This scaling behavior is a little bit much, but you have 2 areas with different scales involved. And so this consistently seems to be the case. What the reason is, we don't know yet. So these are different situations where the male are with males, with mated females, males with mature females, males with immature females, and with fruitless, and you see it's consistently the same behavior of the scales. Now this is particularly intriguing, since we all know that the C. elegans is a scale-free network, and this is so. But if we compare, then we don't really know whether this is true, or whether this is still true here, or whether this is true here at all. So it's not so clear to me under what conditions this holds. And you can also show that the Drosophila courtship graph is a small world network. Now let us switch now. This is just a sort of a caution that all the biological networks should be scale-free. Maybe there are networks that have more than one.
There is a nice question that when the plot that you are drawing, what is plotted there? What are those? Are chemical interactions between different molecules that are activated during...
On the courtship?
Yeah.
This graph is the graph between behavioral elements. So the flies dance on one leg, they wiggle with the ear, whatever they do.
But each of the note of the means a part of the body, or is that...
It's a body language.
So each of the...
So you make a movie of the courtship. Courtship is they dance together, they do things.
Okay, so each of the elements...
You look at the different pictures and you decide what they do in a given moment under inspection. And then you write down the symbols, and then you have a symbolic dynamics.
Okay.
And so this seems that this is a network that connects the different acts, and it seems that there are really two well distinguished parts of the behavior in terms of key. Now the biological data that is probably closer, sorry for not, you were right in asking, it was done in a very brief way, but it was only meant as a sort of signpost. If we go to the biological data, what we see in the visual cortex, we have this orientation maps where these daisies, as they are called, where patches indicate preferred directions of bars for which the neurons fire most when they are presented. Usually you display a picture with bars, and then the neurons react or don't react, depending on the inclination of the bar. And this is what you get, these patterns here. And now we will see that in a better picture later on, if you really see how the connectivity is laid out here in this patch, so from here to where the connections go, the inhibitory and the excitatory connections, then you see that it's really poorly fitted by a Gaussian. And the question is, and this is really a beautiful work done by Ruedi et al., reported in cerebral cortex. And so this is much more that of a power law behavior than an exponential behavior. Now, what is the connection with the two scales I was referring before? This is from theory. We know that these patches that we observe in the visual cortex, in this orientation tuning, could in principle be so-called Turing patterns. And to generate Turing patterns, what is essential is that you have two diffusion coefficients that are not the same. And this diffusion coefficient really defines the transport in the system, and the Bruselator theory really tells you where you have these patches that we seem to observe. And Fred Wolf in Göttingen really was able to reproduce such patterns based on these active media. Now, so what I challenge is the idea that in biology most of the networks would be scale-free, and they seem to have all small world properties, but when you really look at it, and these people have done that and investigated all the given examples of critical networks, and they found out, I think it was Stanley was involved in one of these works, that you can't really make this claim. And so what we then looked at was were networks where the probability of two lattice sites to be connected depends on the distance in two power laws. So you have a probability distribution which chooses a probability distribution that comes from two origins of two distributions. And we will show that these networks here have advantages in terms of information transport and efficiency. So when you look now back at these measurements and do an adaptation of the results, this is what we did. On a log-log scale, what you see are two decay regions here. And when you really look at the decay borders, then you find that this is pretty much the extension of a cortical column. Now, you may say, okay, this is probably not the power law here. It's close to the beginning of the scale, and there are not too many points. So, but that really seems to be a power law. And what will be sort of important later on for the arguments is this line here that is even less clearly justified. We will test the assumption that here we have a self-similar repetition of the structure, that we have here a decay rate again, like we have here, saying that sort of you have these microcolumns, the columns, the hypercolumns, and then between the columns you have the empty space. So it seems, so to say, a reasonable assumption. And actually, the authors of this study, Ruedi, said small fraction of inputs originated more than 1 millimeter away, and he said, and he refused to draw it here. And so this would be a similar decay. At least this suggests the presence of 2 or 3 power laws. We will implement over this distance only an exponential decay. And what we will do, the difference will be little on that small extension, and by doing so we will also be able to directly compare our results to results made by Markram and co-workers. And so this is for the excitatory, and this is for the inhibitory neurons. And so what we seem to have, if you believe in what I said, or if you are willing to accept this as a working condition, we have computation within the columns, within these structures here, neurons that are here, and we have computation among the columns. And one claim by Markram was that, so to say, if we knew exactly how this is wired in the column, we could, in a sense, re-engineer the cortex. This is one basic assumption. And this is then called also the canonical cortical microcircuit. When you really want to know what is canonical, then you run into problems, because these fixed probabilities are not so fixed, and the Pope is the boss of the canonical things, and he is quite fixed, or has been at least in the past. So we will look at these things separately, and we will start with the computation that are done within the columns, where we now try to find out what kind specific wiring schemes are able to contribute to the efficacy of the computation. To do that, we will work with 2 models we will have a three-dimensional grid of 3 by 3 times 15 is about 135 sites where we put the neurons, and then we start to wire them. We have for the wiring two models, a very simple one and a more complicated one, and this really refers to the canonical microcircuit is built up here. The other is much more simple in the sense that here we have a probability to connect, or a weight distribution between excitatory and excitatory, excitatory and inhibitory, inhibitory to excitatory, and inhibitory to inhibitory neurons. The control network is where this is all the same. So here we test whether this wiring really has an influence. And the probability of connectivity we scale as an exponential. As I said, here we use an exponential by the distance, e to the minus distance divided by some lambda, and both squared, where the control network corresponds to lambda equals 2, then the unconnectedness is lambda equals 0, local next neighbor connectivity 1, and global connectivity lambda infinity. And by this we can really check many networks, and the second model that we will analyze is this canonical microcircuits, where we have here the probability to connect here. So this is this connection scheme that we saw here, implemented as a probability distribution on connecting from here to there. And the control network then is that this whole structure does not really matter, and so we can go from this connectivity pattern to this connectivity pattern by rewiring, respecting these boundaries between excitatory and inhibitory neuron pools, and just randomly reconnect, and forgetting about the structure where the connections originally came from. Now, how to compare these different architectures of columnar networks is by means of a reservoir computing scheme, where the signal goes in and is relayed by an input matrix to the reservoir. Then the wiring scheme within the reservoir is in the connective matrix W, and then there is a readout via a readout matrix that is outside, and learning just happens outside the reservoir. This is the important thing, otherwise you will not be able to learn anything without changing the structure, and we want to test the structure. From that point of view, I think that this was a clever setup by Markram and colleagues. So the learning is confined to this matrix here, so we really can test what is going on inside based on the wirings that we apply. And we also use different electrical and mathematical models of neurons. So we will see two of them. We have reported two of them in this slide course here. This is the liquid state machine. Oops, I had it already. Then the neuron models that I will report results of are leaky and fire neuron dynamics. And here are these two matrices. This is the reservoir neuron matrix, and this is the readout matrix. And they also plug in here for the fast spiking simple which neuron dynamics. You may say, and of course this is expected, that the neural model does not really play a lot of influence, but you never know. So you have to check a simple one, an integrate and fire neuron, and a more complicated and realistic one. In both neuron models, the synaptic signal is characterized by an exponential decay to be really realistic. Here the synaptic signal decays in this way here with a characteristic time scale. And it's very important for the results to really care about that you compare the same things. When you rewire, or we do several realizations of the same scheme according to random selection of the wiring following these schemes, the wiring schemes, and therefore it is really important that after each wiring you really check that the same activity is obtained. So what we do is to scale input and synaptic efficiency so that they can be excited by the presynaptic partners, that something is happening even without input from outside. And the firing rates, however, needs to be such that we are sufficiently distant to saturation, otherwise we cannot distinguish between different results. And then we also need to check the connectivity matrix W to be of comparable strengths, and this we did by scaling the common largest eigenvalue to a particular number, so that they all have the same behavior. And these are the results. Now we have two tasks, real-world tasks. Markram et al. Really looked at it for more theoretical tasks, where they said that there was an advantage if you wire as in biology. Here we choose the Auslan sign language, which is a rather complicated one, and then the Arabic digits speech recognition task, which is a simpler one. And the reason for doing that was we wanted to see whether the system would at least react upon tasks that it could do or could not do. This is why we choose the Auslan sign task. And the other task should be doable. This is why we chose this one. For this task, you can make the calculation that you have not enough neurons. So this is really A and B. This is A and this is B. Then we have the blue curves for the leaky integrate and fire, and the red curves for the sequence neurons. You see at one glance that it doesn't really matter what you have. Then what you also need to see is here in this first model, where you have only the probabilities between the connectivity of excitatory inhibitory as in nature, compared to the same model where the probabilities are all the same, the control network. This is the control network, so are the dashed curves. Again, what you see immediately is that the dashed curves are on top of the other curves. So there is really nothing in that distribution. What you can also see is that here we have a sort of this behavior, and you can really understand this behavior, but I will not comment too much on it. How the connectivity model has an influence in it. Important really is that the control network and the original and the really explicit networks fall on top of each other, here as well as here. Here we changed this parameter that defines the neighboring the neighboring wiring, and here what we change here is you could really think that you could really think that the inner wiring of the neurons in the reservoir must be of a certain type to have hidden neurons, in a sense. And this is a parameter that we changed in order to go from all of the maximal number of hidden neurons to none of the hidden neurons. And what you see is also that even this does not really matter in the present context. These are these variables here. And also here you can argue, you can really understand why this increases, but this is another thing. Then here, for the more complicated neural network, the more detailed one, we again have these two tasks that we look at, and once again, and this P is the rewiring probability. So initially you had this performance here, and after you rewire, nothing happens. So what you really have is that this is irrelevant as well. I forgot to tell you that we modified the original reservoir network readout by a little bit more clever readout that really pushed up the recognition rate here from 0.5 to 1. So you can really do a little bit by a more intelligent readout that gives you more, essentially more freedom to store patterns. But the thing is here really you have the change in the hidden neurons. Also, it's not that because you lack the hidden neurons that you don't see any change here. But it's really clear that the control network follows the explicit network schemes. Now you could sort of criticize that because we only display averages taken over many network realizations, that it's probably just because these averages are so poor and so widespread. And here you can see that these are more or less Gaussian ensembles these are the distributions of the different realizations in one go, and you can see that there should be a well-defined average. So this is certainly not the problem. And then you could also say, okay, maybe this dependence on the network wiring parameter lambda depends on the number of neurons that you look at. So maybe you can say that in a column we have many more than 135 neurons, and here we show the results from 135 up to 980 neurons, and you see the lines essentially fall on top of each other. They have the same behavior, and so it's also not the number of not the number in the column that really generates this lack of response towards the wiring scheme. And of course here also you can see that the network with the largest number of neurons, of course, performs a little bit. Now, I think it's fair at this point to say that it seems, at least in this reservoir computing scheme, that is arguably the only one that really can test what a particular wiring does in the connection of pattern recognition, because otherwise you always have these learning steps that really change your wiring. I think based on this, you can say the wiring does not really matter in the column. And so that the question is there probably something particular in how the columns connect to columns on that level of the... network. And so we implemented these two scale networks, and again by using these paradigm, we can go from next neighbor or random coupled columns to fractal columns to doubly fractal columns. Here you see this. Here you see these two scales. Not entirely in the sense that I showed to you with the Drosophila graph. There it was this direction, but this is how these double scale fractal connectivity networks would look like. Now, of course, you could now condense a single column to a hyperneuron or columnar neuron, just to project it on one big thing. But what we do here is to proceed a little bit more mathematically by saying that the recognition rate that we would look at before transform in the question of how good such a network is in terms of being able to transport information as quick as possible through the cortical network. So we are interested in the speed of information transfer. And secondly, it's not just enough to transport information somewhere and leave it there, but it really must generate some coherent reaction at the point where it goes to, or at least the possibility must be. So what we look at is how difficult it will be to get enough of this information traveling to a certain point as to, at this certain point, generate a change of the state of the column that we look at. And so, where a temporally coherent computation really takes place. And this in mathematical terms would be expressed by saying these cells there synchronize with the signal that arrives there. And if you want to do that, then you have to model the columnar interaction by means of a connectivity matrix. And now the laws that you have is the transfers in the probability of generating these ones in the connectivity matrix as a function of the distance. So you have, for example, the nearest neighbor connectivity, the Gaussian exponential, and the power law. And if you look at the velocity of propagation, what we see is that the velocity that we observe for fractally coupled networks, this means a power law connectivity, behaves in the following way. The nearest neighbor networks in this way, and you see that the fractal really is much better. And now an important thing is really how many of these connections do you need to generate this change at the target point. Because as you know, in the brain, most material is really wired and not so many cells. So the volume is really filled up with wires essentially. And so the wiring length really is important. And in the first step, we look at the number of connections that we have for different sizes of the network and the velocity that we then have. And for average number of connections 8, this is the nearest neighbor network. The fractal is much better, and so on and so forth. Here we have the saturation regime. And we can see this, how the thing propagates. Here we apply a perturbation on on a network that is on a line. And what we see here is the spread here. And it takes a lot of time to spread for a very little area or distance to be covered, whereas at the fractally coupled network with a decay of 0.6, you see that in a very short time the information propagates a lot.
Let me change the batteries. I think the batteries are not.
So, now how do you model the models? Now the models, they need to deliver spike or whatever, what a real column does. And you can always create a dynamical systems map that creates spikes according to what you want. Rulkov has shown this for many cases, and we applied it in the hearing system. It's really a wonderful tool. You can generate from bursting to regular spiking to everything what you want by using the right parameters. It's a sort of a uniform modeling tool. Now, if you agree that you can do this, then you have a map representing each column. Whatever the column does will be represented by a map. Now you have to define how these maps interact. And these maps, of course, can interact by means of coupled map lattice diffusive coupling. So here you deliver a little bit of your activity to the neighbors and you get from the neighbors. That's the scheme. And now you can see how the perturbation or the information that you have that arrives or departs from a certain point is spreading. The spreading of this perturbation is described by the Lyapunov exponent of the map that you have. Of course, these columnar maps must be chaotic in order to be able to react on changing input and in order to recognize incoming input via the coupling. If they refuse to talk to the neighbors, then you don't have any chance of a reasonable modeling of the situation. Then another important thing is the diffusion coefficient of the Gaussian spreading. And these two things, the instability and the diffusion, they affect a site I at a distance I from the center of the perturbation, given an initial perturbation d0 in the following sense. So the perturbation that is generated is this factor times e to the lambda times t minus this exponent part that contains the diffusion coefficient d. And the velocity that you are able to derive from this expression of in particular the exponent is the velocity is 2 times the square root of the local Lyapunov exponent times the square root of the diffusion coefficient. Yeah.
When you use diffusion, are the columns connected bidirectionally or unidirectionally in the real brain?
Well, in the real brain, of course, they are bidirectionally, but when you send something out, it's one directional.
So I mean, I don't know what will be the conclusion, but this speed and these kind of things for diffusion coupling. I'm just wondering, is this realistic to assume that the columns are diffusively coupled? I mean, it's a model. It's a model, but I think it's a reasonable one.
And this is the velocity of the information wavefront. And now when you look at it, what you can see is that what you will sort of ask for is that each column has about the same instability. So the same build-up dynamically. Otherwise you cannot come to any further conclusion if you don't accept this. So when you now say that this is for all columns the same, and this is just the network characteristics, then you can calculate the velocity just by calculating the diffusion coefficient. That defines the speed of the information wavefront. And the synchronization at the front is given by the eigenvalue of the graph Laplacian in the following sense, that this must be fulfilled for all case to be synchronized, while this is the largest, or the eigenvalues of the graph Laplacian, and this is again the local sitemap, Lyapunov exponent, and this can usually be obtained by increasing the number of connections that you have to the point. This is actually a result that you can look at the papers of Chen. And now the results that we obtain by looking at this are the following. For all, this is the number of, on average, connected neighbors to the point where you start from with the information. You see that the doubly fractal network is, this is the speed of information transfer, is much larger than the single fractal case here. And the random network and the nearest neighbor coupled network. So this tells you that as a function of the number of neighbors being connected, the doubly fractal architecture is by far the most efficient, the fastest, and it seems also in terms of the neighbors the most efficient one. Now, this is, you can ask what's about these values here, whether they are special or not. So if you choose two decay scales that are different, whether these are particular values or not. Here are the results, and whether also how it scales with size of the network. And what you see here is that here, if you go through all different exponents, you observe consistently the same story. Here you have a saturation effect at the end. This is just given by the size of the network that we look at. And here is how it develops with the size of the network, and it's also clear that this is consistent. Now, what you really should look at is for every network topology, you would have to really do this calculation of the graph Laplacian. And of course this looks different, whatever the wiring scheme may be. And so when you really look at this in detail, what you find that in order to have this synchronization at a given speed, you have a smaller number of connected neurons on average for the random network than for the double scale network. But you have a lot more neighbors in the fractal case. But when you also look at the length of the wires that these neighbors induce on the brain wiring, then you find that the doubly fractal wiring scheme takes much less wiring if compared to the fractal and to the random connectivity network. So it seems indeed that the biological neural network that we have optimizes the total wiring length that it needs, given a certain speed it wants to achieve. And this is consistently over all these scales or this area where you do not have a saturation effect. We checked it in many different places and this holds. So it seems to me that we really have a special type of network connectivity in many biological situations, and that it is not a scale-free network that is per se the best solution in biology. And it seems that this Barabashi-Albert scale-free network is, when you look at the spectral density here, then this would be the theoretical distribution for the Barabashi network. This would be the random network spectral density, while our networks here have a completely different form, and I think they are quite distinct. So maybe this is really the reason, maybe the reason for defining a new type of network, consisting of two scales. And this is why I showed you these graphs at the beginning. There, the scale inclination, the inclination of the slopes has a different order than here, but we still need to be to find out whether this is generally true or not, and where it comes from. But it seems to me that these scale-free networks don't really describe the wiring scheme, at least, of the brain very well. They may describe other things very well, and so these excitatory inhibitory power law decay exponents that we saw in this paper of Ruedi, they are really close to those from this critical avalanche model of cortical computation. So I'm not saying that this is not really a good model, but maybe when you look at the wiring, it seems to me that two different mechanisms are involved and not just one. So the conclusions would be that you have worse performance if you consider only a single fractal networks, and this may be at the origin of the increased complexity of the macroscopic biological neural networks, as they have been observed by Kurtz, for example, in this paper here. Then what is worth noticing is that the doubly fractal networks, they achieve this superior performance irrespective of whether their long and short ranges are done by neurons with long and short connections. You could think of a neuron that has preferentially short, or preferentially long, or preferentially short and long connections. And this, of course, you can monitor in the connectivity matrix, and it has no influence. Or what you could think is that you have neurons that have long connections and other neurons that have only short connections, and whether they are organized in a columnar structure or not, whether they are just distributed, also does not matter. The matter is that you have long connections and short connections. And the columnar structures that we observe in the cortex may therefore express a sufficient but not necessary facilitating structure of combination of speed and minimal wiring length. And it's also worthwhile mentioning that most monkeys, carnivores and ungulates do have columns, that's true, but rats and mice don't have it. And so rats are not particularly stupid compared to some carnivores, and so this seems really to support our argument. And then the characteristics of the optimal networks that we have found, the mean path lengths and the clustering coefficients, they are quite close to those of C. elegans and other networks that we know that people have investigated. And this then seems to be the end of my presentation. Thanks for your patience with me. And
your double fractal connection structure and modeling it with a couple of map lattices disregards propagation speeds in the connections, right? Your long distance connections are as instantaneously coupled as short distance ones. Do you think, or what do you think would be the effect of taking into consideration propagation speeds? Or does the model rather refer to local areas, but what would change if I go to the global brain?
no, I think that sort of the time that you need to go there is obviously reflected in the connectivity length.
Is it? No, because it's still instantaneous, the coupling. Even the long connections are instantaneous in your model, right?
Yeah, but the time that you need as a whole to arrive there is till the last has arrived. You can take a model that you would say, okay, we have the smallest wiring, so it will be most instantaneous in a sense.
Because the long-range connections facilitate getting faster coherent structure or synchronized pattern, right? But if these long-range connections are important, they might also be associated with propagation length. So how do, for example, propagation lengths compare to your signal, your information transfer speed? Are they comparable, or how do they relate to each other?
No, so what we did was to fix a propagation speed and then compare the networks that allow synchronization at this propagation speed. This was this plot. And both things are sort of engineered by the number of connections k that you have. And this gives you a network that can achieve the transport and synchronize. And now if you ask how much time does it take to synchronize or to go there, then I would say a guess over the thumb would be the total wiring length. If you have a long wiring, total wiring length, I would expect it to take longer. But it doesn't need to be. But this k is an average number of connections. So I think that is probably the best way of looking at it.
Any other question? I have a curiosity. What is the role of the neuron within the cortical column in that case? If they don't play computational role, what do they do?
They do play a computational role, but it simply does not seem to depend so much on how the wiring is. And of course, if we are looking now on a general purpose column, not a sensory column or something like that is specialized. And we are not looking at a column that has learned to do only one thing. So this is, I think, if you apply learning to it, then it obviously involves what needs to be learned. The wiring will be different. So if there is something in these probabilities in a large scale, then it must show up before you have learned.
Mm-hmm.
Otherwise, we need to have a sort of a learning network, and then the wiring will be different for every task that you have.
Yeah. Any other question? Well, if not, let's thank again.